deck, that is tether all out.
Copy, tether all out.
Those look like mahi-mahi.
What?
Yep, it's a school of mahi-mahi.
Oh my gosh, that's incredible.
Don't tell Mike.
That's so cool.
Oh my gosh, there's a lot of them.
Those are gorgeous fish.
What are the chances that... Mahi?
Control deck, that's Atalanta off the deck.
Copy, Atalanta off the deck.
Yes. Yes, they were mahi-mahi. Yeah. You guys can hear? Or is it just me? Okay.
Yep.
No, yeah, okay, cool.
All of us can hear.
Everyone can hear.
Sorry, yeah, that was really cool to see that school of mahi-mahi. So
No, right?
It was.
I was screaming here.
Noel, did you get the message about the fixing the focus on the wide field camera array?
The one that Dan was hitting it with a camera, with a hammer?
Uh-huh.
I did see that.
So are we running a photogrammetry survey on this?
I think to my knowledge, we can.
Okay.
I can double check that.
If y'all don't mind, I'm gonna do a audio slate, if you guys don't mind.
I don't Same here, Max. No audio from you.
I'm going to do an audio
hear
slate,
you.
if you guys don't mind.
Oh, I can hear you.
Okay. This is an audio slate for dive H2076.
Control winch. We are all stop 50 meters.
Copy. All stop 50.
Are you ready for control?
Pilots are ready.
And
Thank you.
All right, can audio slate again. This is dive H2076. UTC time is 2317. Let me try that again. Apologies. The clock changed. I wasn't ready. Okay.
It's going to keep changing, buddy.
This is an audio slate for dive H2076. UTC time is 231820. Mark.
Yeah. Why don't we save it for then? Yeah. Although we're dropping at 1,150 meters right on the base of a ridge, and it's going to be like tomorrow or yesterday. It's going to be really, really dense. This dive will be deepest about 1150. Yep. Mind if I change that, Lonnie?
Go ahead.
Okay.
Just a gentle reminder, ROV. Oh, no, it's on, never mind. I was about to tell you about ROV upper.
Stuff. Oh, bioluminescent stuff. Oh.
Something drifted by in the camera.
Oh.
Sorry, I should have said that on SPL. I'm going to switch us back to dive salvo, everybody.
Bridge, nav. Can we please make a move back to waypoint 1, bearing about 260? Thank you.
It was a mahi-mahi. Yep.
Okay, I'm going to switch back to dive special here, salvo.
That looks better. ROV team, just checking in. Do we have the green light to start? Awesome. Okay. All right, to our viewers just tuning in, welcome to the 8 o'clock watch. We just launched our vehicles, and then our vehicles are currently descending to our dive site number 7, right, Jeff? Awesome.
That's correct.
Okay, but before we get into it, let's start with our introductions. How about we start from the front? Is that okay? Sure. Yeah,
Awesome.
we can do that.
Okay.
Good morning, everybody. This is Derek Sowers. I'm the Mapping Operations Manager for the Ocean Exploration Trust, and I'm the navigator on this watch.
This is Gabby, and I'm sitting in the Herc seat.
And I'm Rai, and I'm sitting in the Atalanta seat.
Hello everyone, my name is Max. I'm sitting in the video seat and I'm a video intern.
Awesome. Hey everyone, my name is Lottie Halleck. I am sitting in the SCF seat, or the Science Communication Fellow seat.
Good morning. My name is Jeff Drazen. I'm a professor at University of Hawaii and a deep sea ecologist, and I am the science lead for this watch.
Good morning, my name is Piera. I work at the Palau International Coral Reef Center, and I'm currently on the Nautilus as a scientist.
Good morning, everyone. Ali, my name is Noelle, and I am the science coordinator with the Ocean Exploration Trust, and I'm in the data logger seat.
All right, thank you, team. If you want to find out more about what we do on board, about Nautilus, I encourage you to go to our website, nautiluslive.org. It has our biographies, and you can find out more about us on the website. Yeah. So can we talk a little bit about our dive site? What's the plan here?
Sure. So last night we mapped this area. This is we are about 30 miles south of Angar Island in Palau, and we are diving to a depth of about 1,150 meters on the northwest side of a ridge-like seamount structure. So this is actually a very similar feature to, although a little shallower, to the one we dove yesterday. We'll dive down on the northwest side, we'll come up the flank of the seamount to its ridge, and then we'll follow the ridge along to a depth of about 700 meters.
Oh.
So I anticipate, if yesterday's dive is any indication, that we will have lots of coral and sponge garden on this shallow seamount as we go up. And hopefully, again, like last time, a lot of interactions with pelagic animals that happen to be drifting through and hitting the edges of the seamount.
Thank you. Is there a reason why we chose this specific dive site?
We are trying to pick sites all around Palau that represent the National Marine Sanctuary and Palau's waters. And so this site was chosen as one of the ones sort of to the south of the main islands. And it is also relatively close to one of Pickric's environmental DNA sampling sites. But we so far have explored seamounts to the and features to the north and to the east, including the slopes of the trench and seamounts up to the north and to the east. So now we're just kind of moving around to the south of the islands.
Okay. So yesterday you mentioned something about mapping first before we launch the vehicles. Can you tell us why that's important?
Most of Palau's exclusive economic zone is not mapped, and in fact that is true with much of the world's oceans at large. Palau is not an outlier here, that it is quite normal for us to have not stellar maps of large regions of the world's oceans. So before we dive on a feature, we actually need to know what we're getting into. We need to know what it looks like so that we can put the ROV down on appropriate habitat type. So last night, after we ended the previous dive, we used the ship's multi-beam sonar system and drove to this site and began mapping it. So now we actually know what the shape and structure of this underwater ridge looks like, so that we could choose an appropriate location to do the dive.
Awesome. Thanks, Jeff. So this is an open question to the watch. Are there any expectations for this dive? I know the previous watch, what we saw was amazing. It was stunning views. Do we think we're going to see the same? I think we're going to see the same. If the mahi-mahi is any indication of it.
It was a very auspicious start, wasn't it?
Yeah.
What was our bottom depth?
So I'm looking forward to reaching the bottom.
Yeah, I think just looking for me, just some amazing dense sponge and coral forest. Again, like the old growth forests of the sea. I think it's really amazing to look for those.
Did we get a confirmation
Yep.
on what our bottom depth
Fingers
is?
crossed.
Fingers crossed indeed. Oh, Piero, can you tell us a little bit what you do in Palau, your line of work?
Sure. Well, I work at PSCRC, the Palau International Coral Reef Center, and well, we are doing a lot of different type of monitoring at Pickrick. We monitor the marine protected areas, we monitor bleaching, we monitor the fish stocks, really a lot of stuff that you can find on the website if anyone is interested. Currently on the Nautilus, when I'm not on watch, I collect water for environmental DNA. So basically we get water samples either from the surface or from with the ROV at the bottom, and then we filter the water and basically all the organic matters that remain trapped on the filter can be used for DNA purposes. That tells you, it gives you an idea of the biodiversity of what was in the water in that moment. Yeah, so we've been targeting fish and marine mammals, but with environmental DNA you can basically look for any kind of taxa you want. So that's a powerful tool to give you an idea of biodiversity in an area without having to see the animals.
Oh, okay.
Yeah,
Is that a fish?
it was.
Yeah, we have entered the deep scattering layer. This is this daytime layer of mesopelagic animals that you often find between about 450 and maybe 800 or 900 meters during the day. And these animals were closer to the surface at night under cover of darkness when they couldn't be hunted as easily by predators like tunas and swordfish. And now they have gone down here to the darkness to hide out until the sun goes down. So we're going through that layer right now, and I imagine we'll see lanternfish and deep water eels and squids and all kinds of things, shrimp. And that's just before we reach the seafloor and see all the communities there.
How exciting.
I want to see another gulp eel. That was cool.
Another gulper eel?
Yeah.
Yeah, I'd love to see that again.
That would be exciting.
Yeah.
I want to watch that behavior again of them quickly opening and filling their mouth.
Yeah.
I was reading a paper last night about that, which was written in 1989. Last time anybody wrote anything about gulper eels to any extent.
Yeah.
And they suggest they lunge forward and engulf prey with their mouth. And we frequently just saw them with their mouth full right after hitting the seafloor or bouncing off of something. So it seemed more defensive than offensive, but more observations would be great. There goes a jelly on the left.
In the paper, did it mention what their diet composition was? Are they lunging to grab fish or
it
like?
It did, and this was from the North Atlantic, and they mentioned that they had consumed very slow-moving squids, like balloon squids, cranchiids, and some fishes, also some more planktonic organisms, copepods, and then sargassum seaweed.
Wow.
And it was found in the stomachs of 12 different fish, so it wasn't just an accident. So they suggest that they're indiscriminate predators that eat anything they come across. And it's a gorgeous jelly that just went by. That might have been in the genus Paralia.
Do gulper eels have the sort of robust gill rakers that you often see on planktivorous fish?
They do not. Instead, they have a very restricted gill opening at the back of their mouth, and so the water goes out through a very small space at the back.
Very interesting.
But they don't have the really fine gill rakers on the gills at the back that you would see like on a herring or something that filters lots of small plankton. Ken is chiming in because I think Ken knows a whole lot about gulper eels too. So let's see if he sets me straight on any of my quick reading facts. So for those of us who are joining, we also have a series of scientific experts online who join these calls. And right now we have Ikolau Otto from Pickrick, and we have Ken Sulak from the east coast of the United States, and we will probably have many more people joining us in the next couple of hours, depending on their various time zones and when they can join. So thanks to all the scientists for joining us on this dive.
Nav, can you recenter? And you're going too fast for me now.
Oh, ROV team. So I have a viewer asking, is the ship moving while the vehicles are underwater?
Yes, they are. It is. So right now we're moving the ship just to make sure that we're positioned directly over where we want the vehicles to start the dive on the seafloor. And then throughout the dive, we're basically stepping the ship forward or in the direction that we want the vehicles to move over the terrain. So really the ship is the primary way that we move the vehicles kind of grossly into position, and then we fine-tune that with, like Hercules can move around within the distance of its tether from wherever the ship is pulled Atalanta to. So just a reminder, like Atalanta is actually connected to the ship with a large steel cable with fiber optics in it and power. And so kind of wherever the ship goes is where Atalanta gets pulled to many meters below. And then since Hercules is connected with a flexible tether in front of Atalanta, it can kind of move around any way it wants to within the distance of the tether. So yeah, that's really kind of the navigator's primary job, is just to coordinate those movements so that the ship is moving in such a way that we are covering the terrain that we want the vehicles to be able to access.
Oh, okay. Thanks, Derek.
Sure.
Derek, since the tether connects from the vehicles to the ship, how long is the tether usually?
How long is the tether that the team just put on? 30 meters?
30 meters.
Yeah.
93 feet.
30 meters or 93 feet. That's long.
Yeah, so kind of behind the scenes, one thing the pilots are constantly keeping awareness of is where the tether is during the dive, and making sure that the vehicles aren't kind of putting twists into it and making loops with the tether, and kind of keeping it in a nice alignment. So they're always cognizant of like the distance between the vehicles and the orientation of the vehicles relative to each other. So the sort of managing the tether is always in the back of their mind, and part of their awareness when they're following things around the seafloor and moving and trying to meet the science objectives of the dive.
Does it, how often does it happen, like if it gets wrapped up in things?
It kind of depends on how much we're moving around. Like what we try to do while we're going over the terrain is have a pace where we can kind of keep Hercules basically out in front of Atalanta, and Atalanta can kind of keep an eye on Hercules from above. But if we're trying to chase something that's moving along the seafloor, like a fish, for instance, Hercules can kind of go off and get out of that position somewhat, and that's when you start to put some wraps into the tether or the cable. So we just try to keep that managed, and then if we have put a twist in it, we try to undo it before we move on. But like yesterday, we had a dive where we had a wrap in the line that we had to take out, and we were managing some strong currents at the end of last night's dive. But I would say it doesn't happen that often. Usually we're pretty on top of keeping the configuration that we want.
Oh, okay. What is the tether made of? Like what type of material?
I'm gonna have to ask the pilots for that one.
Pilots?
I believe the strength member in it is Kevlar.
And then there's like there's a plastic jacket and several layers of plastic and silicone insulation on the wires. And then there's copper conductors that carry the 3-phase power and fiber conductors that carry the telemetry to the vehicles.
Okay. Thanks, Gabby.
Yeah, I wouldn't have been able to give that answer. So,
I think another question is like, overall with the tether and the vehicles, how are they able to withstand the pressure as they descend to the bottom?
Every component is designed for it. Just very specifically designed for it. Components that can't withstand pressure are put inside pressure housings that keep the air pressure around those components at 1 atmosphere, just like we live at. And things that can withstand it do. The foam specifically is built to be buoyant, even though it gets a lot of pressure on it. So it doesn't compress to the depths that we can take it to. So that's very specifically designed bunch of little glass beads in an epoxy type matrix that's like called syntactic foam. It's used on a lot of subsea instrumentation to provide buoyancy at really deep depths. So yeah, every component is specifically designed to either be protected from pressure or to withstand it itself.
yeah, I think it's kind of interesting to a lot of the wires and can you talk about the fluids, the oil that's surrounding a lot of the components?
Yeah, so there's a whole bunch of components that can stand pressure but can't stand water. So we got wires running all over the vehicle, and copper wire has no problem with the pressure, but you don't want to let any water get into it. We have each of those components in oil,
It's sort of like a sealed chamber.
Yeah, totally. Like a sealed chamber with a slight amount of positive pressure on it, between 6 and 12 psi, that always keeps the water out of the sealed chamber, even if there's small leaks.
And so is that oil essentially not compressible when it's sealed like that? Is that why we do that?
Essentially. Sometimes little air bubbles get in, and obviously the air is compressible. But yeah, the oil is incompressible. The oil's acts like water as far as compression is concerned. It's like having all those items, all those components surrounded by water, but it's non-conductive, so it doesn't, and it's non-corrosive, so it doesn't affect the electronics and the electrical systems.
Starting to get beams on Doppler.
Thank you.
So yeah, I mean, for our viewers, that's something to keep in mind whenever scientists want to put other instrumentation on the ROVs. They have to be depth rated to whatever whatever kind of the lowest depth rating is of a particular instrument that we put on is going to be the limit of the vehicle at that time. So if we put on an instrument that's only rated to 1,000 meters, obviously we can't go any deeper than that. So that's, sometimes when we're testing new technologies or we're working with other partner organizations to test things like we did earlier this year, that can become a consideration. And so we have to plan our dives accordingly if we want to use that instrument. Like we had an eDNA filter system that was really cool. You could put it on the ROV and it would pump water whatever depth we were at.
Oh.
And then you'd capture the particles onto a filter and then process them later in the lab. So that was a really neat system, but one of those pieces of equipment was rated to 1,000 meters. So when we had that on the vehicle, we could only go to that depth.
Oh. That will save a lot of filtering.
Yeah, it's pretty neat. This is just a bunch of different bottles with each, or a bunch of different pumps rather, and each one has its own filter, and then you can decide what depth you want to filter.
Pretty cool.
I think on the course of the whole expedition we did like 16,000 liters of water pumped at depth.
Wow.
That's a lot of water.
Yeah, so you can filter much larger volumes. So basically you're increasing your sample size of the marine environment quite a bit.
And Hercules can go to about 4,000 meters? Is that correct?
That's right, yep.
That's really deep. Thanks, team.
I'll stop at 30 when I'm there.
Yeah, that sounds good. Bring it back home. Send the reset from the USBL line. Is that what you do?
Yeah, you ready?
Yep.
There we go. Okay, I'll stop at 30.
Okay.
Ooh, I see the bottom.
Can I see the slope, Derek? Just the high pack screen real quick.
Yep.
Am I downhill of
No,
Atlanta?
you're kind of on the same.
Okay. Sounds good.
Yep.
What's going to be our direction of travel? Can you turn auto head on?
Yeah.
Yeah, we're going to go
Auto head's
about
on.
145 degrees.
Okay.
And that'll be moving upslope towards waypoint 2. Yeah.
We've already got fish.
Nice.
Oh yeah. Oh, this is very sandy. Some rocks in the distance there. It's another fish.
Okay, you want to turn to look at Herc?
Yeah. Is that our heading, 180?
145.
Okay.
Yeah, so initially the slope is fairly gentle, and then about a third of the way to waypoint 2, it starts to steepen up a good amount.
How does tether look like it's laying? Is that
Looks fine.
okay?
Looks beautiful, actually. Thrilling.
Thrilling.
Yeah.
A masterpiece.
Yeah. Hang it in the Louvre.
All right. Okay, our heading is 145.
Beauty. Do you want to give me,
White
do you
balance?
want,
Yeah.
do you want to set up craft for white balance? Video, are you ready for white balance?
Absolutely.
Sweet.
Do I do craft valve or craft power first? Okay. Oh, okay. Okay. Starboard is still on and everything? Okay. Okay, craft valve. Is there a reason why blue button would not be doing anything? Oh, maybe. Okay, so it should work now? There we go. And then it takes a minute? Sounds good. All right, video. How's that?
It's great. Can I zoom?
Yeah.
And are the ROV porch lights off?
All your lights are on.
It says in the checklist to turn the porch lights off. I'm not sure which lights the porch lights are.
That's going to make it really dark.
Yeah, we tried that yesterday. It did, so you did it with all the lights on yesterday.
If it says it in your checklist, we do have another light called the porch light, and it's not hooked up. So,
Yeah, well, we'll call that good then. That sounds like it's probably it. Okay.
Nothing. You're trying to trick me into saying it. Yeah.
Okay, and you guys are good if the camera goes black for a second? All right, here goes. Okay, awesome. Do white balance on. All right, and okay, 1 sec. Okay, I think we are good to go. We go wide?
Yeah, let's go wide.
Is there something? Another fish?
Fish.
Nice job.
And guys, when you're ready, can we get a Niskin, please?
Yeah.
Thank you. You want to check the fish?
Here, I'm just retracting. Okay.
Niskin 1.
Sure.
Okay, I'm watching. Yeah, that went.
Thanks.
What's the sample number?
095.
Say that again.
095.
Thanks.
Just getting you centered up again. Okay, there you are.
When you're ready, that fish is just off to the right.
Go for zoom.
Wow, it's really dark.
It's a black fish.
It's a black fish, yes. This is different than the ones we saw yesterday.
Here, I'll come down a bit.
It's really spectacular how much light we're throwing and how little you can see the detail.
Reminds me of that gulper eel where it just absorbs all the light.
Yeah.
Yeah. There's actually a paper on ultra black coloration in deep sea fishes. Okay, this is a rat tail. It is jet black. They think that the ultra black coloration in fishes like anglerfishes and that gulper eel that we saw yesterday is about minimizing the reflection of bioluminescent light so that animals that create bioluminescence can't see them.
That's very, very stealthy.
Or they can't see the reflection of their own lures off of their bodies. You can imagine an anglerfish lighting up its lure might give away its position, but the ultra black coloration is supposed to absorb all of that light.
Okay, we happy with this?
Yes, thanks.
Okay, go wide.
DVL reset okay?
Yeah, go for it. What is your strategy here, Jeff? Do we want to get cracking towards the waypoints? Do you want to poke around a little bit here?
I want to look around here just a little bit because there's a lot of fish, and then start moving slowly towards that waypoint.
Okay. You did just say there's a lot of fish, which means we're not going to see another fish for 100 meters.
So I just saw one go underneath the vehicle. It could have been a halosaur.
I saw it also, Jeff. Looked halosaur-y.
Max is a great spotter.
Oh, and there's another fish. It's almost in center
it's
frame.
coming.
Oh yeah.
Oh.
Sea cucumber.
Oh, what was that?
Cucumber.
Oh,
That's
yeah,
a
I got
Headless
it.
chicken monster?
Yeah.
Yep, headless chicken monster.
Go for zoom video.
Okay, if we can get any more zoom on this guy, it's really small.
Do you have any more video?
Yes, you want me to do?
Yeah.
It looks like a rat tail to me.
Yep, it does.
So fast.
This one is small enough, I wonder if we might be able to collect it.
If it would stop moving. Go wide. Can you grab the arm?
Wow.
Yep. What are we doing?
We are going to grab the suction.
Okay.
Is somebody keeping an eye on the fish?
Yep.
Yeah.
Yeah,
Barely.
go ahead and grab that, and I'll grab your cameras and everything.
Okay.
I've lost it.
I've lost
I
it
still
too.
see it.
Okay.
Where is it, Max?
It's like center frame still. And it's just a tiny black line.
Oh, sorry.
Where is it, Max?
It's just north of center frame now.
Got it.
There we go.
Okay. Get yourself out front there. And Max, can you guide me in again?
I just looked away for a second and I lost it.
I got
do you
it.
see it? Okay.
Oh, I see it. Okay, I'm going to give you suction now. There, you got it.
Yeah.
And I am going to land sort of a little ways in its drift path.
You getting pulled around? Yeah.
Do you see it still?
Yeah. It's
Is
like
that
in
it?
line with the suction.
Okay.
It's really, really, really close to the bottom.
Okay. Yeah, I'm gonna land and it's drift past, so get up off the bottom there.
Okay.
Oh, now you're gonna swim. Huh. That's funny.
We can get a shrimp instead.
Well,
Okay, coming in for a landing.
you can always
here.
go for a twofer.
There you are.
Go wide, video. Are you already wide? Where to go? Any sign?
No.
Don't see it.
Oh, no.
Piera?
No, mistake.
Oh, there. It's maybe to your right. Swing right. You might see it. Is that it?
Oh, yeah.
No.
Is
Or,
it?
Oh,
sorry, I
see
don't
it.
wow.
see it.
Yep,
Right
right,
there.
there.
It's in the cloud.
Okay. Okay. I won't try and stick lock the landing this time.
Are you coming in for landing?
I will land, yeah, just to
Okay.
get close enough to the bottom.
Okay.
Go for it.
Suction on? Whoa.
Yes.
Come here. Come here.
Nice.
Yeah. All right.
All right. Is it in the tank? Yes.
It is.
Okay.
That's in container number 1.
Here, just give me a sec.
Fantastic. Thank you very
I hope
much.
it's still in the tank
Nice.
there.
I'm gonna put this away.
Nice job.
Oh, another fish right there.
Yeah.
I do see it.
Oh, looks weird.
What
is that?
this? Okay, this may be an upside down anglerfish, you guys. Or I'm totally wrong. Hold on. Let's zoom in before I start jumping to conclusions.
Go for zoom.
What is this?
Oh, zoom out. Yeah, that might actually have been a Gigantactus. Okay.
Go for zoom again.
It's pretty close to us, I think. Okay. This is not Gigantactus. What the heck is this thing?
When it's in the center, you can push in a little further next time I get it centered up.
Copy.
It's zooming for us. Never mind. Hey, buddy.
Zoom
It's
out,
coming
zoom
right
out.
at us.
It's in the camera.
The files are in the computer.
Yeah.
I believe that was a type of, it is, a type of anglerfish. You can just barely see the lure, but you can also see teeth sticking out the side of the mouth.
This guy
right
knows
there.
how not to get collected.
Oh,
Yeah.
that's very cool. I'm trying to
That's
remember.
very
I want to
clever.
almost say Neoceratius or something like that, but oh, that was very cool. So that is an example of a fish that is really mid-water, but has probably just been drifted into the seamount. This is not, it's not a fish that we normally capture near the seafloor, or see near the seafloor.
Another fish that's also so black. The iris is almost all the way up,
Yeah.
but it was just
Yep.
You see no detail.
Nope. They just absorb all light. It's astonishing. So they actually, in designing these ultra black materials, scientists have actually learned a lot by looking at the skin of deep sea fish to understand how that's
the
done.
suction and just keep it in your hand for a little
Yeah.
while?
Sure.
I have this feeling like we're gonna get
Yeah.
another chance for another
Yeah.
one of those. That flying grab was awesome.
That was
Really good.
vindication for yesterday.
Yeah.
Okay. I'm gonna just pull it out a little bit.
Yeah, that's a great place to keep it.
Okay. I'm gonna lock there. Do you want me to keep the controller in my lap, or?
You can put it down
Okay.
or keep it in your lap. Really, that's up to Jeff,
Okay.
whether he's like feeling very sampley.
I kind of am. We're on a roll.
Oh, you can see that fish.
Nice.
Do you
It's
see it
there.
in bucket?
Yeah.
It's awesome.
Oh, yeah.
Very cool.
Okay, Jeff, we are so ready for the next sample.
Outstanding. Max, you seem to have the eagle eyes, so pick out the tiny little fish at the corners, please.
Sounds good.
So wait, did we just sample a rat tail?
Yes, indeed.
So now you've seen a rat tail
I
sampled.
have seen a
Oh,
rat
yeah.
tail captured with an ROV for the first time in my career.
Heck yeah, that's
Nice work.
It is outstanding.
And
Jeff did start clapping, so that's the level of excitement that we're experiencing.
nice one, right?
Very, very happy scientist back here.
And Jeff, correct me if I'm wrong, but rat tails have been a large focus in your career, haven't they?
Yeah, I've been studying rat tails since I was a graduate student, and that's when I was studying their feeding ecology and energetics, sort of their rates of metabolism and growth and energy use. And yeah, they're an amazing group of fish. There's 300 and something species worldwide. They live in all the world's oceans almost, at depths below about 200 meters, and they extend. The deepest living one goes deepest living species goes all the way down to 7,000 meters on the edges of trenches. And oftentimes, particularly in abyssal environments, they end up being top predators because some of the abyssal rat tails get up to about 1 meter in length, and they're one of the largest fishes there because a lot of the sharks and rays are gone. So they're really fascinating. Fascinating species that probably modulate a lot of sort of community interactions through their predation. And
It's so cool having you here, Jeff. I feel like our viewers are always like,
There's
tell
another
us
one.
more about the fish.
oh, right
It
there.
looks like a rat tail, hey? Go for zoom.
Oh, this is a Bathygadis. See the really, really long fin rays on the sides?
Yeah.
Oh, if we can get a little bit more zoom on this guy and then try to capture him, that
Okay.
would be fantastic.
Push in real
We
quick,
have
and then we do have to actually go
this,
for it.
this may be, I think this is in the genus Bathygadis. Yes.
Okay, go wide. Okay, this is a drifter here, so I'll get in his path. I'm going to give you suction.
Oh, just spooked.
Do you
No,
see him?
no, I got him.
Okay. Still drifting?
He's kind of staying in the
swimming.
same place.
Sort of head into the current now.
Perfectly center frame.
If you're watching on channel 3, you can see the telestrator that Jeff is helping guide the team with.
I'm still up a bit
Yeah.
off the bottom, so
This guy's right next to the seafloor.
yeah. Oh, he's spooky. I can see him. You don't need to circle anymore.
Okay.
Okay. Oh.
Oh, coming right at us. There it is.
Wow, he's spooky. He is.
Oh, so close.
Sorry, that was a lot of sediment. Suction
No,
on.
that wasn't on you. I was trying to fly him in there. I actually think that might be the way. If you just hold still on the.
Yeah. Okay.
Oh,
Here, I'll just stay
still
here.
there.
Pretty spooky though. Could you do me a favor and keep me from going too far off into the cabbage?
Yeah. And is suction on?
No, it's not.
Okay. Do you want me to try to get some?
I'm gonna turn it on.
Okay.
And that'll take care of all that.
And so far, you're fine in terms of cabbage.
Thank you. This one's very spooky, Jeff.
Okay. Oh,
Come here.
so it has these extremely long fin rays, which are covered in mechanosensory structures. So this fish is probably very
aware of what we're
aware of the shear of the suction sampler. But it is also now tuckered out. Oh.
I'm sorry, buddy. This is for science.
Yeah, thanks, friend.
Do we see you?
I do, yes, right there.
Okay, thank you. Can you point the suction down just a little?
Yeah. Just give me 1 second here. I just want to come up a bit. Okay. Yeah, you want like that?
There, perfect. That's great. Oh, wow, that was like very much as close as I can imagine coming. Video, can you give me half zoom?
Absolutely.
You can, why don't you go with the arm there, if you can.
There,
Yes.
there.
Oh.
What?
It's like hiding below the suction. You've got suction.
I know.
I don't, and he like found the one spot where there's no suction there.
What?
I'm a little high up
Gotta
now.
get lower,
Yeah.
lower.
I know.
Can you pick the arm up?
Yeah.
down. There we go. Can you do a little reassessing with Atalanta here?
Yeah.
Thanks.
I'm just gonna.
Oh, he's way up off the bottom now.
That was so disorienting.
That was disorienting.
Yeah, you're still okay with the Atalanta.
Sweet, thank you.
If you feel yourself getting tugged on, I can come down a bit.
Okay, sounds good.
Do you want me to move the arm?
Yeah, why don't you pick it up?
It's pretty high up now.
Hi.
Jeff,
Yeah.
what do you think? You want to keep chasing
It's
this?
so close.
It's so close, but this fish is also very much better at this than we are. We can keep trying. I could see us killing a lot of time this way.
Okay, let's keep going.
Okay.
We
I want
haven't-
to get you all the fish, but I also want to be realistic. We can give him one more go.
Okay.
But
I'll get out.
this one does seem to have the better of us. You have suction.
Come here.
There you
Oh.
go.
That was close. I know, it's like the claw game. Like, it's always going to get you real close to keep you going.
It's like gambling.
Yeah.
Fish gambling.
Yeah.
I never go to Vegas.
That's the county fair, by the way.
Oh,
Did I just get it?
I think you just got it. We'll see.
Oh,
Yeah.
Yeah. Nice.
Okay,
Wow.
I'm taking you off that jar.
Okay.
No, I got it.
Got it?
Let's
Okay.
go.
Thank you, fish.
Mercurid and a Bathygadis.
Nice job.
You guys rock. This is amazing.
Do you want to just give me a better view a little
we
bit,
so-
Mr. Sec? I just want to know where I am.
You want me to go full wide also?
Yeah, oh yeah, that would be great.
So just in
why I
case
can't
viewers
see.
are wondering why I might be so excited about catching a couple of fish with the ROV,
Okay, I'm going to
Very
one of the
much so.
important
Here.
components
Beautiful
of
job.
catching
Thanks, you too.
animals with the ROV is...
Oh.
Uh-oh.
What was uh-oh?
Our screen went blank over here, but
telestrator.
Oh,
obviously
sorry.
none of the main ones did.
Okay.
S- The reason is that, you know, we're doing visual surveys, and imagery is great for surveying communities, but oftentimes it's very difficult to actually confirm the identity of the species you see in the video, and fishes are no exception to that. And by capturing some of these animals, and we just want a few representatives, particularly of things that we have seen commonly in a region, you actually have a specimen in hand that you can identify with existing taxonomic keys, and you can match that to the video that you see, which gives you a whole lot more power in the data that we generate from all of these activities. So it is obviously much easier to capture corals, which just sit there, than it is to capture fish. So capturing these two fish is just really marvelous. We so frequently are identifying them, and we really don't have any collections from a given area. A place like Palau, which really the deep sea is underexplored, just a couple of these animals makes a really big difference in the assessments that we can make.
Jeff, do you want to poke around for more fish, or shall we clean up our toys and move on?
Oh, that's such a hard question.
And by
Yeah.
the way, we have made 0 progress on it.
Oh, Derek, we have made so much progress. You have not been listening to what I've been saying.
I am listening. I'm just talking about terrain progress.
Derek's like,
Sure.
my whole job is to keep you moving.
Let's start moving towards the slope. I really would rather not put away the slurp gun for
Sounds
just the
good.
next
We will keep
10 minutes or
ourselves
so.
totally ready.
There's a fish right in front of you if you keep going forward.
Oh, this is such a good dive.
Jeff, folks want to know why we think we're seeing so many fish in this area.
Well, you know, this is a great question. We've just gotten here, so I'm not certain right now. But just as a general
Noel,
rule,
can you confirm for me what is the highest number that has a fish in it? Is it 2?
Correct.
Okay.
2 has a fish in it.
so one of the general observations that people make, and there are definitely exceptions, is that frequently when you get into very steep rocky terrain where we see a lot of the corals and sponges, these are very current swept areas. They're really dominated by filter feeders, and you don't see as many fish. Now, on yesterday's dive, we saw a lot of fish, but the vast majority of them were those Oreos, and a lot of the and it's possible that they're either eating plankton out of the water column, or they're picking animals off of the corals, or eating maybe even some of the coral polyps themselves.
He's going to step away. Just
Many of the fishes that we are seeing here eat worms, small crustaceans, other things like that, and there are actually probably a lot more of them that are accessible to these fishes in sediment-covered areas like this where we've got a lot of sand.
Super interesting.
And that's just simply because most of these fish, they're not eating corals and sponges. They want another group of animals to prey upon, and those are going to be more common in sediment. That's a working hypothesis. No one has actually demonstrated
look at him, but he's like, I'm coming down a bit though.
I also like to hang out with our snacks, so this just feels like it tracks, you know.
Yep.
There's actually 2 at the top of your screen.
So we have another fish expert on our science chat. That's Ken Sulak. He was just confirming that anglerfish we saw was indeed Neoceratius. And the very black rattail or mercurid that we saw a little while ago, both of us still are unsure, but perhaps Cordophenoides.
Awesome. Thanks to all of our scientists ashore. Super awesome having you with us and having your expertise as part of the conversation. Do we have a sense, do these types of fish live their whole life on a seamount, on a single seamount? Are they moving up and down the slopes? Are they moving between seamounts? What do we know?
Well, it depends very much on the type of fish. So a number of the rattails we think probably are moving fairly large distances, certainly over their lifespan. But some of these fish that seem specifically associated with seamounts, like the Oreos, oh, this is great.
Can you go for zoom video?
They might spend a good portion of their life in a single seamount. That is one of the rattails. This might be Kumba. This is a rattail we saw a lot of yesterday. It's got a really, really long body. I need to get one better look at the dorsal fin, if it just turns for us here, if it has dark rays in the front of it and really tall. Yeah, I think that is probably the same thing. Not 100% sure of that.
The ratio to eyeball size to body size on this one really makes me happy.
Yeah.
I really like
these fish are definitely tuned to seeing in very low light levels and seeing bioluminescence.
I'm not sure I can get you a better shot than this, Jeff.
This is marvelous. Thank you.
Okay, go ahead.
Do you think its eyes are literally bigger than its stomach?
Good question.
I mean, with both eyes combined, probably.
Oh, another big rat tail right there.
Can you be more specific?
On the right. Sorry, our
right there. Can you see it?
Telestrator's still reloading. Back here.
Yeah, I can't put marks on the screen at the moment.
Go for zoom.
It's a pointy one.
Oh,
okay, that's good.
This is the fish. This is the one that's so common in baited cameras. This is in the genus Coelurhynchus. It's got a really pointed snout and ridges on the head and around the top of the jaws. And we saw this species. It was one of the most common fish we observed in baited cameras at these depths. and we don't know what it is. We don't know what species of Coeluricus. Now, in this particular one, I'm now seeing kind of a bit of banding. There's a little bit of dark and light on the back of the body, which may mean this is an adult in a species called Tokiensis, Coeluricus tokiensis.
Cool.
But not sure. This fish is definitely not small enough to collect.
Just imagining trying. That's all.
So
It's just making me laugh.
So am I, Gabby.
It's a nice urchin. We could look at the urchin.
Let's look at the urchin.
Go for zoom.
She's trying to distract me.
Well, I know you're getting ideas.
Have you ever flown a fish directly into a bio box?
No, have you heard of that happening?
No.
I can totally see.
It's just a joke.
I would say about 50% of the pilots I know would try that. Especially if you brought it up and said, I bet you can. Okay, go wide. I have sort of flown like Bathygadis into bio boxes before, because they get all floaty and like.
Maybe if we baited a bio box, we could lure them in there and then close it real quick. Well, I'm pretty excited to report that we've moved about 30 meters.
Thanks, Mom. That's good. Look, he wants to go in the suction sampler. Are we there yet?
My job title, actually, the acronym is MOM.
Yes, it is.
What does MOM stand for?
Mapping Operations Manager.
Absolutely perfect. That's your nickname for now.
Shouldn't have said anything.
Yeah, that's definitely on you.
Are we there yet, Mom?
Yeah, we are. Would you like another step?
I don't think that question was rhetorical.
Yeah, no, actually though, Jeff, do you want another? Do you want to keep moving?
Yeah, let's start moving up slope.
Oh, and there is a fish in the bottom right.
Oh, we need to see that.
Okay, let's hold on a second.
Okay, we've got an eel, probably a cutthroat eel. Synaphabranchidae.
Go for zoom.
They so frequently do that gaping or head shaking or as we get close to them. I really do wonder if that's some sort of a threat display or something.
You can push in further, maybe. And try.
So if we can get any more zoom on the head, I'm trying to see which of the jaws is longer, the upper or the lower.
Yeah, we've got more zoom.
Yeah, we just don't have more pilot skill. We're maxed out.
This guy's got a longer jaw? Or lower jaw? So I think this is Synaphabranchus, and it might be Synaphabranchus oligolepis or Aphanus. That's great, thanks. We can keep going.
Okay, go wide.
Bridge, nav. Could we please do 30 meters bearing 140?
Oh, another fish.
Tucked into the little crevice there.
Oh, yeah, neat.
There it is.
Wait, what is?
I'm going
in the craft.
Cuskeel on this one. It looks like the Cuskeels we saw yesterday.
In the crevice there. Okay,
Yeah.
I see this thing
Go
now.
for zoom.
It's got that sort of translucent.
Not pleased that we're here, look.
Yeah. It's kind of cute.
It is.
Yeah.
Oh, wow. Okay.
This is definitely like peeking out your front window to see what's happening in the yard.
You kids get off my lawn.
And twitching the curtains.
Do we have any more zoom?
Go for it.
Maybe Diplacanthopoma or something like that, but I'm not sure which one. But yeah, I agree, Max. This is the same as we saw yesterday.
Okay, go wide.
Nice.
Jeff, could you just like feel that this was a really fishy area? You seem to know right away.
Well, yeah. I mean, when we were high up, you know, being really high is great for seeing bigger things. And at one point, I saw 3 fish at once. So just knew it was outstanding.
How do we?
Maybe we'll get lucky and see some
I have no idea.
sharks or something like that on this dive. Does anybody know how to get the telestrator to restart on the camera here?
Max, are you telestrator savvy?
I've never tried to use the telestrator, but I can go and poke around.
Where do we go here?
I'd be feeling good with.
I don't
Yeah,
actually know
okay.
this one.
Yeah,
I'm guessing
you're welcome to go
Fingerworks
troubleshoot
is
it.
the name of it.
Okay.
Oh,
Oh,
there
here we are.
Great.
Okay, we're set.
Reloaded. We're good.
You're welcome.
Nailed
Scared
it.
it into submission.
Excellent troubleshooting.
Threatened it with a video tech.
Don't make me come over there.
Turn this ROV around.
Mom will turn this ROV around. Jeff, folks want to know, is it possible the fish are reacting to, or I guess kind of like, what senses do these fish have? What might they be, you know, can they detect metal? Can they detect electricity? Can they, you know, what senses?
I mean, they have the typical senses, certainly the vision, and at this depth, they are probably still seeing a bit of ambient light from the sun, just a very little bit. And bioluminescence. Most deep sea fishes that have been studied have incredibly good senses of smell. They have very large nostrils. They also have a lateral line sense. This is a series of sensory structures that sense vibrations and movements of water. And so that's very useful in dark environments. So, for instance, that second fish that we collected, the Bathygadis, that fish had really, check that guy out, had really long dorsal, pectoral, and pelvic fin rays, and they were all stuck out at right angles to the body in like a radar, forming like a radar dish right around with a head as a center.
Go for zoom.
And that animal, we know, does eat some small animals out of the water column, amphipods and things like that, and so little crustaceans, and so it can probably sense those things swimming without even needing to see them. So this little stocked sponge is Sacocalix.
Go wide.
We know that sharks and rays have also an
fine.
electric
I just was
sensing
having trouble with
capability,
something.
and they can detect alterations in electric fields, such as what would occur with animals moving and contracting muscles. There has been a suggestion that some deep-sea bony fishes might also possess such sensory systems, but I think the evidence for that is a little weak, and it definitely needs some more
Go for zoom.
investigation. But deep sea fishes are very well attuned to a very dimly lit environment. So sometimes that's hard for us as humans to envision because we are very visually orienting creatures. We interact with our environment mostly with sight. And while they do use sight, they have all these other senses as well that they probably use to a greater extent than we do. Oh, there's another one.
Another one, what?
Sponge. We can keep going. Same thing. Sacrocalyx.
Nope.
Yeah, viewers wanting to know about the fish colors. Definitely the dark scales and coloration.
Sorry, when I said no.
Yeah, helping not reflect the light around. Not on this peel.
Say that again, Megan. I couldn't hear you.
Oh, no, just folks wondering why they're dark colored, and refreshing that we were talking about them not reflecting the light. In that case.
Oh, yeah.
Fish.
Yeah, this is... Oh, there's fish. Yeah, it's sort of a...
It's gone.
Pira.
What happened with this one?
Sorry, go ahead.
Sorry, we're getting distracted with a few things going on.
Ship
in the
is
control
moving
room at the
again.
moment, but yeah, I think there was sort of a focus many decades ago on the fishes being very dark in color, simply masking themselves from the ambient light environment, from sunlight, and being black in a very dark environment, of course, is camouflage. But now what we're realizing is that there's a number of fishes which we call ultra black fishes. They reflect less than a couple of % of the light that is placed on them. They're just incredible. They're like the fishes we saw earlier in the dive, the anglerfish.
Yeah, go ahead.
And so this, what we're realizing now is that these fishes are probably trying to prevent reflection of their own bioluminescent light. So this is, I think, most obviously observed in the deep sea anglerfishes. So if you've ever seen the movie Finding Nemo and you know about these, them going down into the deep and meeting the fish with the bright bioluminescent lure and the big fang-like teeth. So those fish are real and live in the deep sea. They're not quite as depicted in that movie, but they do have a lighted lure on their forehead, which they use to mimic some other kind of organism bioluminescing and attracting curious prey items in front of their mouth where they can eat them. But the problem with that is that if they turn on a light bulb on their forehead, then everybody else in the neighborhood can see them if the light reflects off their bodies. So by being ultra black, they don't reflect their own bioluminescence and therefore can remain hidden and use that stealth strategy, which is really fascinating.
I'm going to restart upper video. Do you copy?
Copy.
Classic. Upper's going to go black. Jeff?
Yes.
Shall we clean up our toys? I'd love to put the arm to bed for a little while.
Okay, let's do that.
While y'all do that, we'll
Okay.
give some shout outs around the world. What's up, everybody in Palau watching, and also in the United States and Canada, in the UK and Romania, in Spain
Yeah,
and Germany,
go for it.
Indonesia, Australia. Thanks for being here. If you're watching over on YouTube, also feel free to come over to nautiluslive.org. You can send in your questions there for the team, and we'd love to answer them. Toss those questions around the control room
Sorry, I
and
can look at this better.
let you connect with our team. This is the last dive of Libu's Voyage 1, the first leg of this expedition.
Like this?
We will go into port for just a couple days and then be back out diving again by Monday, Palau time.
That is a big rock in front of us.
Definitely enjoy the seafloor now, and then lots more to
there's
come.
a great coral coming up on the left.
Do you have comms? Did you press the blue button? I mean,
We're coming right up on a rock. Yep.
yeah, I see it. Thank
Okay.
you. Yeah.
When we come up here, if we could move a little to the left, there's a coral in the distance on top of an adjacent boulder.
You can see it in the bottom camera.
Couple little chrysogorgids on this boulder.
Yep.
Dead bamboo on
I
the
gotcha.
right.
Thanks for calling that.
Is upper cam,
It's
Yeah,
back.
you did great.
is upper cam here to stay now?
No. I'm going to reboot the whole thing again.
Okay.
Because the upper cam's still choppy. So rebooting the individual camera is not doing the trick. Rebooting the bottle does seem to, though.
Okay. I'll write that down.
Yeah. Thank you.
Love the shout outs.
Why is it not moving anymore?
Oh, they just rebooted it, Pierre. So
Oh,
you can
okay.
see they turned the cameras off. It'll reset.
Okay.
Super cool viewers writing in that they're watching, wearing their 15-year anniversary merch. Thanks for celebrating with us. Really happy to be in our 16th season of Nautilus Live streaming. More than that of OET in existence. It's a cool little stick coral.
Can
Go
we
for
zoom?
zoom?
Bridge nav, 30 meters, 140. Thank you.
Okay, a little whip coral here. If we look at the base, we might be able to tell whether we've got a bamboo here or something else.
You can push in further.
Yep, it's bamboo.
Oh, I see the little segments on it.
Yep.
Okay, go wide.
So,
We get the ship moving again.
yep, maybe Lepidopsis, but I'm not sure.
Yeah. There's a fish coming.
Oh, great.
I don't
Fish
know
coming,
where that is. though.
center left. Are we zoomed out?
We're full wide.
Okay.
I don't know.
There's the fish right there.
Okay, I'm back. What did I miss? I didn't miss a...
I'm trying to remember, Lottie, exactly when you stepped out. We caught a second fish.
Nice.
Yeah, a little Bathygadis, and we are now kind of coming up against the edge of the flank of the seamount. We're starting to get a little bit of steep here, and we're about to head up the ridge.
Okay.
Okay. Okay, we are in a ship move right now.
Yes. Our pilots just swap seats.
Choppy, eh?
There's a fish there.
Yeah.
If it gets too distracting for you, we can switch back to the
yeah.
regular dive salvo.
Okay.
Until then, I'm going to do what I can to troubleshoot with Dan.
Okay. Sounds good.
Something in the water column.
Yeah, go for zoom.
Okay.
And just watch
What the
Delta
heck
because
is
I'm coming
this?
up a bit.
Okay.
Can we zoom out?
Yeah, zoom out. Where'd it go?
It is gone.
Oh,
Oh, there it
right
is.
there.
I
Alright.
can't tell what kind of fish this is.
It's swimming like it just learned.
Rude.
That's.
It's,
I thought that was really cute, actually.
It's so close to us here. I'm coming up a
Okay.
bit.
Okay.
Not sure. Maybe some kind of cuskeel, but I'm not really certain on that one. We'll have to review the video.
Okay. I didn't, yeah, I've lost it now. Oh, there.
Oh,
Oh, he's swimming fast and up. All right.
yep. Let's
I think
keep
I might have
going.
lost it, so. All right, coming back down.
Fish.
Yeah, right in front of us, right?
Yeah.
Uh-huh.
Rattail.
Oh, great rattail.
It's not much on
Yeah.
focus.
There we go.
Oh.
Yeah,
Okay.
go for zoom.
What kind of fish is that?
It's a rattail.
Oh,
It
rattail.
has the same general body shape as a number of the other ones we've seen. The tail on this one is unusually pale compared to the others we've seen. It could be a different species. Oh. Looked like it was actually going to turn its belly towards us. You can see the little chin barbel sticking off underneath. Oh, this is great. Yeah, this may be similar. There's a little jelly in there. This may be similar to the other ones we've observed, probably in the genus Kumba.
There's another one.
But not certain. Okay, Impera sees in the distance another fish coming.
Go wide?
Let's go wide on this.
Oh yeah, I see. I see what you're talking about.
Just give me a heads up when you're planning on going down.
Okay, I am coming down a bit.
Okay.
Just so we're...
Can we get the lasers on?
Yeah.
Yes, you can.
This way, right?
And another one on the left.
Yeah, he might be moving too fast for me, but we'll take a look. Do you see it anymore?
Straight ahead, a little up in the water column.
Yeah.
Okay. Oh yeah, is he in lower? Oh, there we go.
Oh. Okay, this is another cutthroat eel.
There's two of them.
Oh,
Or
rat
two
tail. There's a
things.
rat tail and a cutthroat eel. Rat tail in the back, cutthroat eel in the front.
Okay, go for zoom.
So many
Try to get them both in this shot. Which one's more interesting? Which one you want?
Cutthroat eel.
Okay.
Rat tail's the same we were just looking at.
Okay.
This cutthroat eel has weird white patches on the back of its head.
I might be checking on the cabbage a bit over here, but
Yeah.
It almost looks like it has a fungal infection on the side of its body. It's very bizarre. Do we have any more zoom, or is that it?
you got more video, or is that?
We do.
Okay, yeah, keep going. I'll try to...
Okay, we're going to try to basically see where the dorsal fin on this animal ends, going up towards the body.
You want to look more at its tail?
No, that's good, right there.
Okay.
Great follow, right?
That is really nice. Okay, great, we have what we need.
Okay, go wide. Yeah, nice. Okay, cabbage looks good. I'm coming back.
Do we have enough rhubarb for a pie?
Oh, I did not quite get far enough to check the rhubarb.
Next time.
At some point, we should probably also stop and take a rock sample when we get to one of these rock outcrops.
You want the other fish first?